Ibrahim, take it away.
Yeah, hello everyone. Ibrahim Olashukwu is my name, a postdoc in Cultivars lab. I'm going to be, I understand the launch is pretty close, so I promise I'm going to keep it condensed. I'm going to be building on the foundation formidably laid by Dr. Karen in our earlier two presentations. So I'll be sharing with you the observations regarding the phenotyping of 11 of the 13 cultivars of interest in the PIP-CAP project. And as a background, I'm not going to be repeating, but this is quite essential. I link this up also. The one base pair insertion that was found in Fronteras, which is the GA20OX for gibberellic acid biosynthesis gene that was found in Fronteras and not found in any of the cultivars, which was what essentially informed this effort of phenotyping. The first instruction we got from the genomics group was to compare that to Monterey, which we did. The work the paper is in draft as I speak. And our observation is actually that Fronteras indeed had quite similar number of primary stolons compared to Monterey, but Monterey actually had a very significantly higher secondary and tertiary stolons compared to Fronteras, which drives home the point that the base pair insertion that was found in Fronteras actually made a kind of a great shortcoming for the cultivar to produce runners and indeed daughter plants. So going forward, we went ahead to phenotype the remaining 11 cultivars of interest, and all our plants always start from tissue culture, and they were rooted in the growth chamber. They were transferred thereafter into this hanging gutter system, and those were made to produce daughter plants, which were rooted again and returned back into the gutter system for the experiment. So I would say that if that is called a generation O, as the generation for tissue culture, then what we used for the phenotyping study was the first generation plants from tissue culture. So 11 cultivars, and these are their classification based on day type. So the experiment lasted 12 weeks, and each of the cultivar had 30 mothers, and the design was completely randomized. And we fertigated them using the 3-parameter system. So we utilized a method of coding the daughter plants, which was developed by Sam Humphrey, a former student in Ricardo's lab, and we did modify this method a lot. So it was a lot of work phenotyping, coding about 7,000 daughter plants, and within a space of 12 weeks. So I'll just walk you through how we interpret this code, as that was actually used in interpreting the results going forward. So the 131 here refers to the primary stolon, while what you have here is the secondary, which emanates from the primary stolon, and the tertiary stolon, which is coded here, actually shoots off from the secondary stolon. So this one refers essentially to the first mother of this, of this cultivar, for example, let's say radiance. So this one means the first mother, this 3 means the third stolon on this mother, and this 1 means the fourth node on this particular stolon on this mother. Then on the secondary stolon side, we have the first secondary stolon, then the first node, but this first secondary stolon actually emanates from the essentially this node, this stolon of the primary stolon, right? The third stolon. So then moving to the tertiary stolon where the daughter plant is located, we interpret this to be the first tertiary stolon on this particular primary stolon, and of course on this particular secondary stolon, then second and node, which produces the first daughter plants on this particular candidate's daughter. So this coding actually informed us on how to relatively seamlessly extract our data going forward to determine all the data collection of interest that I'm going to show in a jiffy. So this again, I want to explain that this particular data plant formed on the second node is what most cultivars actually present, which we call the alternate node producing this data. It's a bit different when we see a data plant growing on what we call the mid node, which I'm going to show also. So on the mid node side, like I said, this is 2, which means if we have a stolon growing as it is from the very crown of the mother plant, let's say we have node 1, node 2, node 3, node 4. Normally what most cultivars present is that the daughter plant is formed on the node 2, and the next mother plant, the next daughter plant is formed on the node 4. So like an alternate thing. So you usually have a vacant node in between 2 nodes. Right. So those are the normal scenarios that we call the daughter plant that are formed on the alternate node. But in some cases, we have daughter plants growing right on the maybe if you have one daughter plant, the very first daughter plant growing on the third node, meaning you have the first node, nothing is there, the second node, nothing is there, then on the third node, you have a daughter plant. So those are the kind of daughter plants that we refer to as the daughter plants grown on the mid node of that particular stolon. Another scenario that we see in some cultivars is that even you have, instead of this 131, you see something like 1, 1, 1, such that on the tertiary node, you see this the first tertiary stolon. Then on the first node, on the first tertiary node, you have the first daughter plant. So all these are scenarios that we count as daughter plants that grow on the mid node, as against those that grow on the alternate node. Then these are the terms of some of the parameters actually that we collected in the study, and yeah, the results. So speaking about the primary stolon, so we had that radius indeed had 37, approximately 38 primary stolon per mother, while Albion had the least. I'm going to be showing the pictures also, at the end of the study, like a fact sheet. So, whereas in terms of the secondary stolon distribution, also we had within this 12 weeks of observation, the same radius actually pulled the highest, even almost doubling the number of the primary stolon that it produced, while Moxie had the least. Then the tertiary stolon also went, pretty much presenting radiance as the highest producer. So eventually we had radiance producing the highest primary stolon, the highest secondary, and the highest tertiary stolon, while we had a lot of values in between that follow in terms of the tertiary stolon. Then what we call the daughter plant that are formed on the alternate nodes. So radiance again, of course, had about 357 daughter plants, formed on the alternate nodes per mother within these 12 weeks of study, while Phen, Albion, and Moxie actually produced the list. So on the mid nodes, actually, like I explained what we meant by that earlier. So radiance, in spite of producing the highest daughter plants on the alternate node, still produced the highest number of daughter plants in the mid node, while we had Portola, Brilliance, Phen, Cabrillo, Albion, and Moxie producing the least compared to radiance. Then cumulatively, in terms of the total number of daughter plants that were found on each daughter or on each mother within these 12 weeks of study, Radiance, of course, produced almost 400 daughter plants per mother. I'll show you the picture. And Phen, you see how dense it is and very fragile it is as well. Phen, indeed, and Albion and Moxie produced the list, and these figures are actually significantly different statistically. So talking about the number of branch crowns that were found per mother plant, radiance and moxie actually had the highest of three branch crowns, the branch crown apart from the mother crown itself, right? So then brilliance didn't produce any, brilliance and Chandler didn't produce any branch crown other than the main, crown that supports the mother. Yeah, so the length of the primary stolons also varied in the study, statistically, as we had radiance, Camarosa, Chandler, Sensation, and Albion, appearing to be the longest, in terms of the primary stolon length, while Moxie, appeared to be the shortest in our study. Then the crown diameter, that were tracked on the data plants at harvest. Actually, we had Phin and Albion producing the highest number of crown diameter of daughters at harvest. So we also analyzed the daughter plant internode length, along the stolons of the strawberry plants of the mothers, and we had it that the longest, the internode one. So this data is actually talking about the daughter plant on the alternate nodes. It's not about the mid nodes, because that's really complicated actually. Because the mid node, sometimes, like I said, we have 3 internode length, and in some cases we have only 1. So we reserve that data. If you're interested, we can share that separately. But this particular one, because of the regularity of how the daughter plants grow, we have only 2 internode across all these daughter plants category. So that's why I'm presenting this data essentially on the alternate nodes daughter plants. So Sensation and Albion appear to produce, the fairly widest internode length 1, while in terms of the internode length 2, the same pair of cultivars actually had still the widest length of internode length. So something like a fact sheet that summarizes the whole of the study for viewers. So this is radiance, the one that produced, the highest primary stolon, secondary stolon, and tertiary stolon. You can see how dense they are. These are the 3 mothers that we, experimented with. And at the end of 12 weeks, this is what it looked like, very fragile. Even in the process of disentangling them, they break off so easily. I wouldn't know what gene could be responsible for that. Those are some of the things that would inform our next step in this experiment to correlate this phenotyping data with what is known in the genomics group to be able to unravel whatever genetic mechanism or gene that regulates these observations of ours. So this is Camarosa, which follows very closely radiance, with producing 12 primary stolon and total number of daughter plants, about 200, of those. And Chandler, which is fairly known to be dense also in terms of production of daughter plants, which had about 200 daughter plants as well, with 13 primary stolon. And Brilliance, which also had something similar to what Chandler produced, 13 primary stolons in the 12 weeks of experiments, and almost 180 daughter plants altogether, but without any branch crown production. This is Portola. So Portola also, was fairly leggy, appearing or growing similar to what is known for Sensation. Then, of course, followed by Sensation, which produced fairly 150 daughters in the study, and Cabrillo, also a day-neutral cultivar, producing 14 primary stolons and fairly more than 30 daughter plants in this experiment. Then Ruby June, which is not as long as, in terms of the primary stolon length, compared to the earlier cultivars, but still producing about 120 dollar plants in a trial. Moxie produced a bit fewer number of dollar plants, about 80 dollar plants in our 12-week study, and really reasonably shorter primary stolon length. But something interesting about Moxie was the production of certain exudates, some latex material on the stolon that we really don't know what could be responsible. It's a bit gummy. I don't know what's inhibitory factor that could pose on the dollar plant production, but it's something we noted in the experiment. And Phin producing close to the number of what Moxie also produced, with 10 primary stolons and 13 secondary stolons. And Albion is actually the last on the list, with 72 dollar plants and 7 primary stolon, 16 secondary, and only 2 tertiary stolon. We can see evidently as leggy and less condensed dollar plants production that it actually presents. Then we do conclude that we have a fairly robust morphological database now to interact better with the genomics group, to be able to perhaps unravel the genes that could be regulating some of the observations that we have reported. So huge thanks to Mark Hoffman, Ricardo, Abigail, Bob. Has been a very good reason I've been keeping my job for the last 2 years. Typing 7,000 dollar plants has been very tough, but she shares the pain with me. Kim, so thanks so much for all your help, and the whole members of the Hoffman lab, Ricardo Hernandez lab, and the micropropagation unit that supplies us these tissue culture materials every time we need them. They never said no. Thank you so much for your attention. I'm happy to take questions. Thank you.
There are several questions.
I have one on the crown diameter of the daughter plants. Is that the average for each daughter plant, and your smallest average was 7? Because those are pretty robust daughters.
Yeah, they were, but that's the average to start with. That's the average diameter of all the daughter plants that we harvested in mother. And, yeah, they were quite big.
I wonder if that's a product of how optimized this system is, because even when we were harvesting from like the Cubao nursery from my other experiments, it was like 7 closer to the biggest that we
Like you see,
7 or 10 and
it's a precision greenhouse, and
I think we pushed those plants quite a lot. You see like the numbers that we could use. So if you take all of them, they have like the chunky ones. They throw statistics a little bit.
I've seen the nursery. It's like, yeah, but that's interesting to see it quantified like that, because those are really high quality plants.
Jared.
Yeah, are you sharing? I'm assuming you're talking the breeding programs.
Eventually we will, yeah. So those are just new data. We just got them like 2 weeks ago, right? Yeah.
I was wondering how this data squares with what the practical experience is in the nurseries.
Right, I'm wondering about that too.
When you have a 4 or 5-fold difference in daughter plants, does that translate to what they're seeing at the field level?
Yeah, I'm wondering, I think there are 2. So first of all, we push those plants, right? And I like listening to the guys this morning, they sound to be a little careful about pushing too much because of disease, right?
Yeah.
So we pushed them all. I think we got 400 plants on radiance. We pushed them a lot, right? And then I think for us, I would like to talk to Karen a little bit more, see what if we find genetic differences, and then all of that will be shared with breeding programs. I mean, that's the whole problem. Yeah.
It'd be interesting, without sharing this with them, to get some of the nursery growers to rank these same cultivars in terms of what they think their runnering capacity is, or, you know, they have very precise yield
I agree.
numbers over multiple years. So they could give you a ranking, you know. And
And
it'd
see
be
if it
really
matches,
interesting
or- yeah.
then to compare those.
Right, no, that's a really good idea, I like that idea, yeah. Yeah, especially on the numbers of the dollar plans, yeah.
Before the next one, just, I mean, you guys have to lick that because you could create a major efficiency in breeding program.
Yes,
And
exactly.
mocks, I mean, my guess is, from my experience, Joe, is this is pretty much spot on, and if, but the middle ones are a little bit hairy,
Yeah.
right? Like Moxie, we can't grow Moxie.
Yeah, Moxie is like, I talked to them like Moxie especially because I felt like when Moxie came out, I was like, this seems to be good cultivar, but there's nobody in the nursery grow it, right?
They won't grow, you
Yeah.
won't plant it.
Yeah,
Well, and that's another piece of this is, at what point? Cause I remember having this conversation with the nurseries when I put together that review article. There's a point at which it becomes uneconomical to grow it. And unless it's gonna produce a certain number of daughter plants, it's basically dead
right,
as a commercially viable cultivar. It might have many other wonderful traits, but if it doesn't produce in the nursery, it doesn't produce daughter plants that have a certain number, then it's out.
yeah. Can you go to the dollar plant just quick? There you go, one more. Yeah. Yeah, so I don't know, it would be really good to see, if, I mean, I know about Moxie, right? It's a low runner, right? And we don't have Monterey here, but Monterey is a big runner. It's good, yeah.
Radiance is very high.
Great, yeah.
That's, those extremes make sense,
Yeah.
it's all the middle.
Middle ones, we don't know, right? Yeah, no, that's a really good idea, I like that.
Well,
idea.
the Fronteras was the one that was a little off, it seemed like, right, you were saying it was a low runner. But I think
I mean,
it's
it's not the best,
it's not
but-
awesome, but it's not
It's not a loser.
It's
And we
kind
might,
of in the middle or a little bit above
yeah.
average.
Yeah, I think the biggest, like, when I talked to Les Canyon like a couple of years ago, the biggest issue they had in Monterey was that the organs are shaped this weirdly. And then it's hard to root them, like they have to push them sometimes into the ground, right? But they didn't talk about the numbers, right? They talked more about like, there's some extra with this sometimes.
Mark,
Yeah.
I just say from my experience growing Albion, Monterey and Fronteras in Ohio. For comparison, is that we do also see that Monterey gives us the highest number of runners and the highest number of daughter plants, and then Fronteras is a little bit higher than Albion, but Albion's sort of our lowest in terms of number of runners and daughter plants over our 3 years of
All right, cool.
And perhaps the source of the planting material could also be a factor, as these are pretty much closer to the early generation of tissue culture.
Yeah, well, we were standardized, so everything is standardized still, right? Yeah. All right, so I don't know about you guys, but I'm hungry. Let's dig in. Thank you, Ibrahim. Thank you much.